Welcome to Chapter 7, Microbial Biochemistry. In this chapter, we go over organic molecules, carbohydrates, lipids, proteins, and using biochemistry to identify microorganisms. Introduction. The Earth is estimated to be 4.6 billion years old, but for the first 2 billion years, the atmosphere lacked oxygen, without which the Earth could not support life as we know it. One hypothesis about how life emerged on Earth involves the concept of a primordial soup. This idea proposes that life began in a body of water when metal and gases from the atmosphere combined with a source of energy, such as lightning or ultraviolet light, to form the carbon compounds that are the chemical building blocks of life. In 1952, Stanley Miller, 1930 to 2007, a graduate student at the University of Chicago, and his professor Harold Urey, 1893 to 1981, set out to confirm this hypothesis in a now famous experiment. Miller and Urey combined what they believed to be the major components of the Earth's early atmosphere, water, methane, hydrogen, and ammonia, and sealed them in a sterile flask. Next, they heated the flask to produce water vapor and passed electric sparks through the mixture to mimic lightning in the atmosphere. When they analyzed the contents of the flask a week later, they found amino acids, the structural unit of proteins, molecules essential for the function of all organisms. And as a side note, if you have further interest in looking into prebiotic hypotheses, I would recommend starting with the RNA world hypothesis. Section 1. Organic molecules. Learning objectives. Identify common elements and structures found in organic molecules. Explain the concept of isomerism. Identify examples of functional groups. Describe the role of functional groups in synthesizing polymers. Biochemistry is the discipline that studies the chemistry of life, and its objective is to explain form and function based on chemical principles. Organic chemistry is the discipline devoted to the study of carbon-based chemistry, which is the foundation for the study of biomolecules and the discipline of biochemistry. Both biochemistry and organic chemistry are based on the concepts of general chemistry, some of which are presented in Appendix A in review. Elements in living cells. The most abundant element in cells is hydrogen, followed by carbon, oxygen, nitrogen, phosphorus, and sulfur. We call these elements macronutrients, and they account for about 99% of the dry weight of cells. Some elements such as sodium, potassium, magnesium, zinc, iron, calcium, molybdenum, copper, cobalt, manganese, or vanadium are required by some cells in very small amounts and are called micronutrients or trace elements. All of these elements are essential to the function of many biochemical reactions and therefore are essential to life. Now this figure is based on mass. Hydrogen is the most abundant element, but it doesn't weigh very much. By mass, carbon is the most prevalent in your average yeast cell, which is what is pictured here. The four most abundant elements in living matter, carbon, nitrogen, oxygen, and hydrogen, have low atomic numbers and are thus light elements capable of forming strong bonds with other atoms to produce molecules. Carbon forms 4 chemical bonds, whereas nitrogen forms 3, oxygen forms 2, and hydrogen forms 1. When bonded together within molecules, oxygen, sulfur, and nitrogen often have one or more lone pairs of electrons that play important roles in determining many of the molecule's physical and chemical properties. And again, you can see Appendix A for a review. These traits, in combination, permit the formation of a vast number of diverse molecular species necessary to form the structures and enable the functions of living organisms. Clinical focus, Part 1. Penny is a 16-year-old student who visited her doctor complaining about an itchy skin rash. She had a history of allergic episodes. The doctor looked at her sun-tanned skin and asked her if she switched to a different sunscreen. She said that she had, so the doctor diagnosed an allergic eczema. The symptoms were mild, so the doctor told Penny to avoid using the sunscreen that caused the reaction and prescribed an over-the-counter moisturizing cream to keep her skin hydrated and to help with itching. What kind of substances would you expect to find in a moisturizing cream? What physical or chemical properties of these substances would help alleviate itching and inflammation of the skin? Well, later on we will go to the next clinical focus box. Meanwhile, living organisms contain inorganic compounds, mainly water and salts, again Appendix A, and organic molecules. Organic molecules contain carbon, inorganic compounds do not. Carbon oxides and carbonates are exceptions. They contain carbon but are considered inorganic because they do not contain hydrogen. The atoms of an organic molecule are typically organized around chains of carbon. Inorganic compounds make up 1 to 1.5% of a living cell's mass. They are small, simple compounds that play important roles in the cell, although they do not form cell structures. Most of the carbon found in organic molecules originates from inorganic carbon sources such as carbon dioxide captured via carbon fixation by microorganisms. Organic molecules and isomerism. Organic molecules in organisms are generally larger and more complex than inorganic molecules. Their carbon skeletons are held together by covalent bonds. They form the cells of an organism and perform the chemical reactions that facilitate life. All of these molecules, called biomolecules, because they are part of living matter, contain carbon, which is the building block of life. Carbon is a very unique element in that it has 4 valence electrons in its outer orbital and can form 4 single covalent bonds with up to 4 other atoms at the same time. See Appendix A. These atoms are usually oxygen, hydrogen, nitrogen, sulfur, phosphorus, and carbon itself. The simplest organic compound is methane, in which carbon binds only to hydrogen, as in this figure, and then here's a bonus figure, a 3-dimensional conformation of methane. As a result of carbon's unique combination of size and bonding properties, carbon atoms can bind together in large numbers, thus producing a chain or carbon skeleton. The carbon skeleton of organic molecules can be straight, branched, or ring-shaped. Organic molecules are built on chains of carbon atoms of varying lengths. Most are typically very long, which allows for a huge number and variety of compounds. No other element has the ability to form so many different molecules of so many different shapes and sizes. Molecules with the same atomic makeup but different structural arrangement of atoms are called isomers. The concept of isomerism is very important in chemistry because the structure of a molecule is always directly related to its function. Slight changes in the structural arrangements of atoms in a molecule may lead to very different properties. Chemists represent molecules by their structural formula, which is a graphic representation of the molecular structure showing how the atoms are arranged. Compounds that have identical molecular formulas but differ in the bonding sequence of the atoms are called structural isomers. The monosaccharides glucose, galactose, and fructose all have the same molecular formula, C6H12O6, but we can see from this figure that the atoms are bonded together differently. Isomers that differ in the spatial arrangement of atoms are called stereoisomers. One unique type is enantiomers. The properties of enantiomers were originally discovered by Louis Pasteur in 1848 while using a microscope to analyze crystallized fermentation products of wine. Enantiomers are molecules that have the characteristic of chirality, in which their structures are non-superimposable mirror images of each other. Chirality is an important characteristic in many biologically important molecules, as illustrated by the examples of structural differences in the enantiomeric forms of the monosaccharide glucose or the amino acid alanine. Many organisms are only able to use one enantiomeric form of certain types of molecules as nutrients and as building blocks to make structures within a cell. Some enantiomeric forms of amino acids have distinctly different tastes and smells when consumed as food. For example, L-aspartame, commonly called aspartame, tastes sweet, whereas D-aspartame is tasteless. Drug enantiomers can have very different pharmacological effects. For example, the compound methorphan exists as two enantiomers, one of which acts as an antitussive, dextromethorphan, a cough suppressant, whereas the other acts as an analgesic, levomethorphan, a drug similar in effect to codeine. Enantiomers are also called optical isomers because they can rotate the plane of polarized light. Some of the crystals Pasteur observed from wine fermentation rotated light clockwise, whereas others rotated the light counterclockwise. Today we denote enantiomers that rotate polarized light clockwise, so a plus direction, clockwise is the plus, as D forms, and the mirror image of the same molecule that rotates polarized light counterclockwise, minus, as the L form. The D and L labels are derived from the Latin words dexter on the right and laevus on the left, respectively. These two different optical isomers often have very different biological properties and activities. Certain species of molds, yeast, and bacteria, such as Rhizopus, Yarroia, and Lactobacillus species, respectively, can only metabolize one type of optical isomer. The opposite isomer is not suitable as a source of nutrients. Another important reason to be aware of optical isomers is the therapeutic use of these types of chemicals for drug treatment, because some microorganisms can only be affected by one specific optical isomer. Biologically significant functional groups. In addition to containing carbon atoms, biomolecules also contain functional groups, groups of atoms within molecules that are categorized by their specific chemical composition and the chemical reactions they perform, regardless of the molecule in which the group is found. Some of the most common functional groups are listed in this figure. In the formulas, the symbol R stands for residue and represents the remainder of the molecule. R might symbolize just a single hydrogen atom, or it may represent a group of many atoms. Notice that some functional groups are relatively simple, consisting of just one or two atoms, while some comprise two of these simpler functional groups. For example, a carbonyl group is a functional group composed of a carbon atom double bonded to an oxygen atom. It is present in several classes of organic compounds as part of larger functional groups such as ketones, aldehydes, carboxylic acid, and amides. In ketones, the carbonyl is represented as an internal group, whereas in aldehydes it is a terminal group. Macromolecules. Carbon chains form the skeleton of most organic molecules. Functional groups combine with the chain to form biomolecules. Because these biomolecules are typically large, we call them macromolecules. Many biologically relevant macromolecules are formed by linking together a great number of identical, or very similar, smaller organic molecules. These smaller molecules act as building blocks and are called monomers, and the macromolecules that result from their linkage are called polymers. Cells and cell structures include four main groups of carbon-containing macromolecules: polysaccharides, proteins, lipids, and nucleic acids. The first three groups of molecules will be studied throughout this chapter. The biochemistry of nucleic acids will be discussed in Biochemistry of the Genome, which is Chapter 10. Of the many possible ways that monomers may be combined to yield polymers, one common approach encountered in the formation of biological macromolecules is dehydration synthesis. In this chemical reaction, monomer molecules bind end to end in a process that results in the formation of water molecules as a byproduct. And that brings us to the end of Section 1. Join me next time for Section 2, Carbohydrates.